So I want to start I'm going to start with Dr. Ramsey.
Okay.
And so I'm going to ask everybody the same question at the start. But my first question for Dr. Ramsey is, what is the core question that you've been trying to work on through the grant that CureGRIN has funded?
Thank you. Right, so the title of the grant was blood cells as a source of potential biomarkers for GRIN disorders. And really the big question is question 2, can we find biomarkers? But also question 8, which symptoms are due to receptors outside the brain, because what we were wanting to do was study the blood of mouse models and also patients with GRIN disorders. So I think that pretty much sums it up, yeah?
Okay, and so next for Ian, can you answer the same question? What is the question that you're trying to answer with your research?
Okay. Is there a slide going to pop up?
Actually, maybe while we're waiting for that slide, I'll go back and ask Amy, can you tell us more about what a biomarker is?
Yes,
and
okay.
what we can use it for?
Okay, great. So a biomarker is some kind of an indicator of the state of the disease. So it would be something like, this indicator is in this range in a healthy person, and it's in this range in a person who has the disorder. So we've probably all had a blood test at some point, and something gets flagged, like maybe you have low ferritin or low iron, and they say, oh, you have anemia because this indicator, this biomarker, is changed. It's outside of the range of normal. And blood is such a great part of our body to measure, because it really doesn't hurt that much, and everyone can give blood, and it doesn't do much to the body to lose a little bit of blood. And we're very grateful for everyone who has donated and asked your children to go through that process. So thank you so much for that. But what we want to do then is to study in the mice and in people, is there anything that stands out that we could use as an indicator? So this is comparing people who have GRIN variants with people who don't, and see if there's a difference that we can detect. And that's really useful for clinical trials. It's useful maybe to help differentiate for a patient if they're having more of a gain of function or loss of function variant. But really our goal was for clinical trials, that it would be really nice to have an indicator, that's like, okay, their iron is low, now we gave them iron supplements, and look, their iron is back to where it should be, so the iron supplement worked. But the same thing, but for treating GRIN.
Thank you. Another kind of biomarker that people sometimes talk about for neurodevelopmental disorders is also EEGs. Mm-hmm. And so maybe it's possible that if there's a variant that is likely to develop epilepsy, where patients are likely to develop it, a treatment before they even start might be able to prevent it, and you might be able to see a change, for example, in the EEG before those. So the medicine's working without a kid having a seizure.
Right. I think the nice thing is that if it's something besides a rating scale that's a survey, it just gives you more of a number that is less susceptible to subjective things that are very hard to compare between how one person would rate it and how another person would rate it. So if it's a number, then everyone can agree that this is the value, and there's less of that interpersonal variation,
Okay. So Ian, what's the core question that you're trying to answer?
Okay. So what we're interested in looking at, I will just wait for the slide again so I can remember which order I wrote this down. So a key question we're interested in is, are drugs that directly target AMPA receptors, can they be effective against the variants that underlie GRIA disorder? So we know from work with GRIN and work with GRIA and work with GRIC that the variants that underlie the disorder are often caused by variants that produce either too much activity or too little activity. And also what we're beginning to learn is that the evidence suggests that if you have a drug that can directly counteract the effect of that variant, they have a better than even chance of being effective therapies. But for AMPA receptors, aside from the inhibitor perampanel, there are no selective drugs in the clinic that you can use for AMPA receptors. And so what we're using is electrophysiology, and there's some examples at the bottom there of the kind of results that we get to test a range of drugs that either boost AMPA receptors, so these are PAMs, positive allosteric modulators, or can inhibit AMPA receptors, NAMs. And we're looking at a range of different molecules to see if they can be effective against counteracting the effect of the variant. And so in the bottom left there, you have some loss of function treatments. So there are some specific PAMs that are currently in development, and we've had a look at those to see how well they work against variants. There are also some very old PAMs as well that were developed many, many years ago, but were never actually approved, but are still potentially there as safe molecules that could find a use as in GRIA patients. And there's also a few little small molecule options as well. And in general, if you look at that first line of electrophysiology, you have the healthy receptor in black, you have the unhealthy receptor in orange, and when you apply one of these positive allosteric modulators, and then it's all overlaid at the bottom, you can see that the green trace looks more like the black trace now than the orange trace. So we're kind of trying to work out which of these molecules are able to do that to make the receptor look a bit more like normal. And then in terms
Ian,
of...
can I just interrupt for a second? Because this might be the first time that a lot of families have seen like that kind of... So what are we looking at when we see those squiggles? And,
Sure. So you're quite right. Sorry, I just get so used to looking at them. I forget that not everybody is an electrophysiologist as their day job. So basically, the way that we look at these receptors is to try and make them think that they're at a synapse. And at the synapse, in general, they sit there and there's no glutamate at all. And when a signal comes along, they suddenly see a really high concentration of glutamate. And that binds to the receptor and causes an ion channel to open, which causes a little electrical signal. And what you're looking at there with those colored traces is the shape of that electrical signal. And where the flat line to start with that's just the membrane doing nothing. When you apply glutamate, it goes downwards, and that's the electricity passing through the channel. And then very, very quickly, it turns itself off. So AMPA receptors are incredibly quick, 1 millisecond. 1 millisecond in general, they kind of close. But this variant here, this I654T, it closes faster than it should do. So even when it's activated at the synapse, it's not producing a big enough electrical current. And so this is the kind of this is the strategy that we're hoping we can contribute towards getting one of these drugs into the clinic someday, whereby you have a drug that is able to make the variant receptor look a lot more like the healthy receptor, and that's kind of what we're building towards. And for the left-hand ones, we're looking at loss of function variants that aren't open for long enough. For the right-hand ones, we've got a variant that stays open too much. So this is one of the gain of function variants. And what we're hoping to look at for these is, of course, we're looking at perampanel. It's our only trick. It's like, are you gain of function? Take perampanel. Are you loss of function? No perampanel for you. And that's all we've got at the moment. And trying to increase the range of potential things we can offer to GRIA patients is really what's driving this project. And so perampanel, yes, we're going to check that and see how it works against these different types of variants. And also we're going to look at some other classes of inhibitor drugs to see maybe those could find a use as well in the clinic. So.
When you, not everybody was in the session that you gave on Thursday,
Sure.
And you talked there about gain of function and loss of function, and it was interesting the way that you talked about like kind of the low level of activation and the high level of activation.
Sure.
And what happened. Can you go through that for everybody?
Yeah. So in general, the way that glutamate synapses work is that they're able to pass signals really, really quickly all around the brain. And if those signals are quite small, then you only really activate the AMPA receptors. And when the AMPA receptor signaling goes wrong, that's obviously a problem. But if you have a lot of activation at that glutamate synapse, a lot of AMPA receptor activation, that's when the NMDA receptors kick in. That's when the GRIN receptors kick in. And you end up with a much larger signal. And if you have an AMPA receptor that is overactive, a gain of function, it's going to recruit NMDA receptors more often than the brain really wants it to. And if you have a loss of function AMPA receptor, even when that signal is the first neuron is really telling the second neuron to go, it's not going to be able to produce enough excitation to then recruit the NMDA receptors. So the way that they work, it's a synergistic relationship. They really do rely on each other. And so another thing we're considering is the idea that drugs that work for GRIN may well help work for GRIA, because this key part of their physiology overlaps. And vice versa, that you could have a drug that affects AMPA receptors that would have a better than even chance of helping patients with GRIN disorder. And so by building up this basic information, looking at the way that the drugs directly modulate healthy receptors versus variant receptors, we're trying to build this kind of almost like not quite a database, but to try and get a longer list of potential candidate drugs that are going to be useful for both GRIA and GRIN, but obviously focusing on the GRIA to start with.
Thank you. Yeah, so those receptors, the NMDA and AMPA, I guess kainate and delta are a little different, but NMDA and AMPA are really closely connected, and so is CAMK2. It's involved in that process as well. And at the end of today, there's a session talking about the interaction between all of those receptors and proteins.
Yeah, and I must apologize for GRIC and GRID patients. I talk a lot about GRIA and GRIN. The majority of patients here are GRIA and GRIN. GRIC does many of the same things that GRIA does, but it's got a kind of a more varied role, and we at least in our lab, we kind of, it makes it complicated to think about GRIC because there's a whole load of other stuff it's doing. It's nice to think about that synapse where the signal just goes from one cell to the other and it's all traveling in one direction. When you start thinking about GRIC, there's some, certain kinds of feedback, and they're kind of found in different kind of places. So so apologies for glossing over GRIC. You're very important to us.
Okay, so over to Dennis now. So Dennis and Ian and Amy have we're our first round of grant funding. So we'll start with you. I know that So the same question for you, Dennis. What's the core question that you're trying to answer?
Yeah, so let's see what slide shows up. so a little bit, let's see how that goes.
Maybe you're using the blue muffins.
The different types of blue, yeah, but okay. So, what we are doing is super easy. We just think that everyone is a robot, and if robots work based on simple mechanics, this is maybe the different slide, but okay, perfect. So, and if you figure out what is broken, you can fix it, right? So, and I really believe that we are all robots because every night I tell myself, tomorrow you don't eat this chocolate breakfast, and then I'm standing in front of it. All my willpower is there, but I cannot do something against it. And this is 100%. We are robots, guaranteed. But the good thing is, if we are robots, is if we understand the puzzle pieces and how things will interact and work with another, we can try to fix things. And so that's what our lab is doing. Is we are trying to work with experts like Amy, Ian, and many scientists here in the room to build, to take all the data which is generated and try to connect it and to understand the construction plan of the robot, how, if you build this part, how that interacts with the other part, and so on. So we don't do anything ourselves, but we're trying to collect and trying to connect things. And so, for example, here's the GRIN portal, which was mentioned earlier, and we're building other tools. So our group is the so-called dry lab. We're just sitting in the office, drinking coffees, eating cookie, and then we're waiting until people generate data, and then we try to connect it and try to communicate this. And here's also, like, for example, our genetic counseling service, where I think they are the only one which have in the US access to 130 million people in the US, where we can do in 12 states in the US genetic counseling, and we are calling it precision genetic counseling because we have all this data, right? So we have a lot of knowledge and a good network to experts, and experts are including families, right? You guys have more knowledge about patients than any physician has, and we can then sometimes ask questions. And when we do, for example, a genetic counseling session, we can integrate data, a little bit of research knowledge, and can try to move into the area of prognosis, right? So typically people talk more, this is the reason why your child is sick, or while you're sick, but we are trying to move a little bit more in the direction, hey, yeah, this is probably the cause, this is probably what's happening to you molecularly, and this might happen the next years. And we can then write into the electronic health records, because we write really formal reports, hey, maybe you should get a movement disorder assessment for your child the next years, every year. And sometimes it's good if you do formalize this, because then other doctors who might not be familiar with the disease, if they read it, it comes from another medical center, they act sometimes more on this, and you are not dismissed. So this is what we're doing. We're trying so basically to understand what is broken by collaborating with a lot of scientists, and then try to implement this clinically. And the question, what is so with this big picture, and we're doing a lot of things. The specific question, what we answered with the grant which was funded, was can we understand how the NMDA receptors or the GRIN genes work, and take all the functional data which has been generated, because there's a lot, and understand the rules, and maybe we can understand how the AMPA receptors, so the GRIA genes work as well. And this is what Ludovica is focusing on, because the NMDA receptor looks very similar to the AMPA receptor. If you think about this, one which you would say, hey, this is one maybe a Dodge truck or pickup, and then you have like a Ford pickup. Both are pickups. They are not motorcycles or other kind of cars. They look really similar if you would be a biologist. And if something looks similar, it often functions similar. And we just need to figure out what are the nuances, and maybe we can take all these 200 functional tested variants and learn our lessons from that we can also understand function of mutations in the GRIA genes without having to test all of them. And this is a so-called prediction model. So we try to predict what the function is, and then at the end this might help patient enrollment in clinical trials, because you think, hey, you are more like gain of function-ish, and you probably you could get that drug, which helps with down-regulating, or you more loss of function-ish, it helps with up-regulating, even if there's no functional data available, because we may be able to make educative guesses. And the tips are clinical trials, and also for prognosis, because people with different underlying broken things, right? So have sometimes different directions of how the disease goes. And this is what the project is.
So people like, just to make sure that I understand, so people like Steve Trinellis and like Ian, they kind of do the detailed functional analysis, right? So they know what the variant is, and they go into the lab and see what's actually going on. But are you using kind of like an AI? Is it AI to see, like, if this is what's going on with the ones that we know about in the lab, what do we think will happen with these ones that haven't been tested in the lab yet?
Yeah, so this all fits into the major category of AI. If you want to get a lot of money, you would call it AI. But at the end, it's not that complicated. So basically how it works is, if in your Dodge pickup, the radio is broken and you can still drive, then we think, hey, maybe if the radio is broken in a Ford pickup, maybe you can still drive. However, if in the Dodge pickup your engine is broken, maybe, and you cannot drive, maybe if your engine is also broken in the Ford pickup, maybe you can still not drive. It's because both engines are important. And this is the type of hypothesis which we are testing. And Ludovica showed like 2 days ago that in principle it works, and we have to figure out more details, because broken is different than driving too fast or too slow. So these gain and loss of function ones is what we are focusing now, and we are working with a lot of collaborators, and we are all very open to work with more collaborators on this to learn this jointly.
Okay, so it can kind of help families, clinicians, researchers, drug companies understand both which variants are actually pathogenic, if they're currently variants of uncertain significance, and to know if it's gain or loss of function, and so which kind of medicines would work better.
Yes, so that's the goal, and we are hopeful, and because there's examples from completely other diseases where this has worked. Everything comes always with a certain level of limitation, but at the moment, so far so good. We're moving in the right direction. We're actually very hopeful that we can do this with certain level of caveats for many variants, maybe even all. But at the end, for clinical trials or prognosis, you always want to have multiple lines of evidence. So prediction is one thing. If there's variants functionally tested, it's even better. And if there's some clinical evidence that your child is more one version of the disease or the other version, this is even better. If you can take a lot of information in, that one can then, as a team, make an educative guess.
Thank you. So, Dr. Dallman, same question that we've asked others. Can you tell us what the core question is that you're trying to answer?
So, yes, waiting for the slide. But the core question that we're trying to answer relates to GI symptoms. So what is the population norm for GI symptoms in people with GRIN-2B? So we've been funded for a project to look at GI symptoms in GRIN-2B. And we've developed okay. And then second question is, can we effectively benchmark GI function in people with GRIN-2B? And so this is somewhat similar to the idea of a biomarker, having some kind of objective measure that is easy to do. So that's where the blue muffins come in, where we can send blue muffins to families so that they can try to have their family members eat the blue muffins in the morning after an overnight fast, and then look for the blue color in the bowel movement. And that gives a time for movement through the gut. And then what is the underlying biology that causes GI distress in people with GRIN-2B? And in that case, we're utilizing a zebrafish model that Howie Sirotkin at Stony Brook University made. But we're looking at GI physiology in that model. And the reason that we chose zebrafish is that the larvae are transparent. So you can look at the gut that is, you feed the fish, you watch their guts process the food. And you can do that in pretty sophisticated ways by actually looking at the cells of the gut that are involved in signaling and processing the food. And so then you can ask, is the gut moving as you would expect? If not, how is it different? Is the signaling, so these genes are all receptors, so signaling is really important. Is the signaling that underlies normal digestion operating as it should?
So we know when we do the Greek census and in other studies that GI issues are a very common symptom across the GRI disorders and really across neurodevelopmental disorders. Do we know why in general, or is that really what you're trying to answer?
I think that's what we're trying to answer, but I think there's been real progress in better understanding what the GI symptoms are across neurodevelopmental conditions. And also, there's an understanding that some of the underlying causes are likely to relate to the GI tract itself, as opposed to just function in the central nervous system. And so many of these conditions, the focus has been the central nervous system. But the periphery is also really important for function of these genes. And so getting a better sense of what these genes are doing in the periphery is super important for managing these symptoms. But I would say we're right at the beginning, because just as the brain is a big place, the periphery is a massive place. And so we're both looking at movement and also at nutrient sensing, because those things are very important for how the gut functions and how the different parts function together.
Yeah, I wasn't in the CVI session yesterday, but I heard from families that there was a lot of talk about how CVI symptoms can impact, so that's cortical visual impairment, how that can impact so many other things that kids are doing, like that their ability to even walk is affected by how well they can see. And it seems like there's also kind of an effect with GI issues. Can you talk about that?
Yeah, so we've done a pilot study and with using the blue muffins and also tracking symptoms over time. And one of the things that tracking symptoms over time reveals is that, I think as everyone knows, there are flare-ups where the GI symptoms can be more severe, and often associated with that may be increased aggression or sleep problems. And so there are many things that go along with gut discomfort. And anyone who's had any kind of interoceptive discomfort knows that it affects mood significantly as well. And so yeah, there's a big interaction between the body and how you feel.
Thank you. Okay, so Dr. Penn, can you tell us what the question is that you're trying to answer?
Right, so we've got that weight again for the slide, so.
We really rely on these slides, don't
Yeah,
we?
yeah.
I just don't remember what I put in them.
Well, everything I've got is pictures, so I'd like to describe the pictures.
We were originally going to put them all into one deck so it went smoothly, but some of the slides came in later, so we're scrambling a bit.
Yeah, well,
No names.
Who do we blame for this?
I think Andy was early with his slides.
Thank you very much. Thank you. Brilliant. Yeah, this is the slide I wanted. So the kind of key question that we sort of are trying to provide some proof of concept as for an answer is really, could the implications of functional classification of GRIN2B variants depend on age? And so if you have a look at the slide, on the left-hand side, we've got what a typical receptor that's encoded by the GRIN2B gene, how it may function. So if we use this sort of traffic light analogy, these receptors opened thanks to their binding to glutamate, as Ian was saying. But it should only be open really for when it's supposed to be. And it's that Goldilocks idea that Keith was mentioning yesterday. It shouldn't open too much or too little, so just in between. And as you know, there are multiple subunits, multiple GRIN genes, and another GRIN gene is the GRIN2A gene. And actually, the receptors that are formed, the subunits that are formed from these genes can actually assemble into the same sort of receptor complex. And the bottom of that left-hand side shows an example where you've got one copy of the protein from the GRIN2B gene and one copy from the GRIN2A gene. And that affects the function a little bit, but within sort of normal parameters. Now, on the right-hand side, what we're showing is a GRIN2B variant, and you can see that the channel is functioning in overdrive on that sort of the left part of that of the variant. And we can see that the little green trace that I'm showing above it is it goes up and it stays up for a long time. Now, some work that we did in my lab and published a couple of years ago was that actually if you've got a copy of the protein from GRIN2B and GRIN2A in the same receptor, then that can actually affect the way that that variant behaves, okay? So we can see on the right-hand side, if you've got the 2A in there, then actually the same variant can behave very differently and not function in overdrive, but in fact, it could either function more like the wild type, or in this case, I'm showing one that's functioning less than sort of the typical receptor, I should say.
So maybe it's just important to remind people that when the NMDA receptor comes together, there's always the two proteins that are created by GRIN1, and then there can be a combination of others that are sometimes 2A, sometimes 2B, most common, and then there's the other ones as well, the 4 other ones that could also be part of that. So you're talking about if it has the GRIN2A, it behaves differently than if it's 2 GRIN2B.
Yeah, and I've actually not shown GRIN1 on here, just to try and avoid...
but we can imagine there's always 2 GRIN1s
There's always 2 GRIN1s, yeah. And the really interesting thing about this is that actually during development, the how much GRIN2A is expressed, the typical GRIN2A is expressed, increases, right? So we can imagine that early in development, a particular variant that is considered gain of function might appear how it is with the green light, too much on the green light when we talk about traffic lights, and that later on in development it might behave either normally with the GRIN2A, or it might even be too long on the red traffic light, right? So we are really trying to understand, provide some proof of concept whether the changes in GRIN2A that really happen during development are actually capable of changing the way that these variants, how they behave.
And so at different periods of child development, there's different amounts of proteins that get made. So the genes are always there, but there's other factors that kind of tell which proteins to make from the genes. And so how does that change? Like around what age does it start shifting from GRIN2B to more GRIN2A?
So in humans, it's sort of late embryonic and early postnatal. But so that's the steepest change. But actually, there is work that indicates in humans that GRIN2A expression increases gradually, even with aging.
Okay.
Right? So yeah, you can think of genes like chapters in a book, and you read the chapter with GRIN2A later, as well as whereas GRIN2B, you read it all your life.
Okay.
Yeah.
Okay. And so. So this could mean that, and maybe this is something we're getting to in your next slide, but basically, because I think we get attached to finding out if we have gain or loss of function, and then think that that's what we have, and those are the kinds of treatments that could help our kids. But you're saying that it could actually change.
So yeah, that is my next slide.
Okay. Okay.
But yeah, but yeah, that moving forward, that's what we would like to try and address. Yeah.
Okay. Why don't we keep things simple for AV and move on to the next question then for you, Andy. We'll stick with you. Yeah.
Okay, so the next slide, please. Yeah, okay.
So we're not leaving everybody in suspense.
Yeah. Oh, I feel privileged to go straight to my next slide. So, essentially what this could mean is right, you might be told what variant you have, and that a lot of the functional validation is done where there's 2 copies of the GRIN2B, right? And so, you're really only kind of looking at how that receptor would look and behave early in development. And we think that actually that functional classification might change a bit later in development when GRIN2A is expressed. And what this could mean is that maybe we want to treat patients differently at different stages of development, and that might depend also on their variant. So if we have a look on the left-hand side where we've got the typical GRIN2B receptor, if it's functioning normally, as I showed on the previous slide at the top, but if we apply a drug like ridipridol, it means that the channel is more on the red light, so less current is flowing through the channel. And so that's not a good thing to give the typical receptor, right? A typical person, it wouldn't be a good thing to give them ridipridol. But if your receptor is normally sort of in overdrive, it's too much on the green light normally. If you look on the on the right-hand side where we've got the variant, and you've got where it says ridipridol, ridipridol could correct the overactivity in the NMDA receptor when you've got the 2 copies of the GRIN2B. But later in development, when there's the GRIN2A, you might want to think about a different treatment, right? So we're looking, everything that we do in my lab is very kind of proof of concept, and in fact, my last slide will go into that anyway. And so it's really to provide some kind of proof of concept of whether the types of changes in the levels of GRIN2A, how much that chapter of GRIN2A is read at those sort of stages of development really makes a big enough difference to make considering different treatments something we need to think about more.
Okay.
Yeah.
So looking at your slide, you talked about this before, but about the kind of development switches. But the GRIN2B, we can see that getting starting to fade, I guess, after or just before birth. And that's when the GRIN2A kicks in.
Yeah.
And then it looks like up 20 years or so, it looks like there's much more of the GRIN2A, right? So
Yes.
So, I mean, we don't know. I guess a lot of what your work is doing is to try to figure out if there's a switch and when that switch is. But in theory, it could be like kids under 10 might be, what would it be, loss of function, and then after 10 could be gain of function?
So the hardest thing, so everything we do will be in a cell system.
Yeah.
And so what it will give us is ideas in terms of the development of neurons that are growing in a dish.
Yeah.
So extrapolating that, these sort of relationships between the neuron development in the dish and the human development is something that is harder to pin down. And I think, I think other work that is that people are doing to look at sort of the developmental switch actually in humans, the timing of it, the precise timing will be beneficial to actually helping move this forward, right? To try and apply this more. Yeah.
Okay.
So GRIN2B, it looks like it's fading out, but it doesn't fade out completely. GRIN2B is expressed later in development as well, just not to the same extent as earlier, and it's more the GRIN2A that's coming up. Yeah.
Okay. Thank you. We'll move now back to Amy for the second question, which is how your work will lead to, or actually, I don't know, maybe we didn't get to that part of your question, Andy. Can we go back to Andy and just talk maybe more explicitly with this slide? How will it lead to treatments and cures, or how could it lead to treatments and cures, your work?
I can comment on this a little bit in my last slide as well. So
okay. Well, we'll wait then.
We'll wait, yeah.
We'll keep people in suspense for that one.
As you wish.
Hey, yeah, go ahead.
Okay. All right. So how will finding biomarkers lead to treatments and cures? I think that there's two ways. One, if we find a biomarker that can help us distinguish, between is this a gain of function or a loss of function effect on the receptor, then that can guide the type of treatment that we have available to us right now. And then, so for example, with these other things, would a certain type of medicine be appropriate or not appropriate? What I'm really most hopeful for is that, because I think the functional studies are going on in other ways, but I think finding a biomarker would help with being ready for a clinical trial. When there is a medicine, we're probably going to need to find a company that's ready to do a clinical trial, and before they would take on this big job of doing a clinical trial, they would want to know that this trial has a chance of being informative, that it's really we're going to get a definitive answer, yes, this is working, or no, it is not. And many times the answer comes back no, but you wonder maybe we didn't do it the right way. And so if you have these biomarkers, then it gives you confidence that, okay, if this is a good biomarker, then it will tell us whether the drug actually did what it was supposed to do and worked, we were giving it at the right dose, we were doing all of these things, or, we did all those things and it just doesn't help. So I think that's what I'm ultimately hoping we can do by studying the blood.
Okay. Yes, early on, as I was working on a podcast about our family, my son Bryson, and our search for a diagnosis and then for a cure. And Amy was one of the people that we interviewed for that podcast, so she's in there. But as part of it, we also, my wife and I went to Boston and met with a number of biotech companies there, and we were sort of asking them, like, what are the most important things that we need to do as a new organization that really wants industry to come in with treatments and cures? And one of the things they said was like having cell and animal models for the disorder. And so we were lucky that we already had some of those. But another one that a lot of them talked about was having biomarkers. So when we came up with our research roadmap, one of the 10 essential questions that we had was identifying biomarkers. So this is one way of looking at it. We talked about the plasma study that we've been recruiting for. So Amy's team can have access to it, and then the industry-funded study can have access to it. Combined Brain, through this industry-funded study, is also going to be launching an EEG repository So we'll be coming back to the community later this year, asking to help work with you to get EEG samples in there to look for biomarkers in the brain.
Yeah.
Thanks, Amy. So over to Ian, back to Ian. And how will your work lead potentially to treatments and cures?
Well, I guess kind of the obvious ways by identifying potential drugs that could directly counteract the effects of variants. But that's a bit more. There's almost, it's difficult to put a time on that, because if the drug is approved, we want to be ready to know that it's going to be good for these patients, good for those patients. We're not so much in control of when that molecule may happen. So we're looking at potential new drugs, but we're also looking at potential drugs that could be used right now. And so that's that side of things in terms of new treatment. But in terms of what we can do right now, what we're understanding more and more about these variants is there are many different ways in which the variant can break normal AMPA receptor function. And so my top line there, we've got at least 7 different ways where we look at a variant and the key mechanism as to why that receptor has gone wrong. There are multiple different ways. And actually, of course, I should actually add there is at least 8, because there is a subset of variants, and they've come up recently, where in our experiments they don't seem to do anything. But the genetics of it make it basically certain that this was pathogenic. So there are many, many different ways in which the receptors can go wrong. And so what we're trying to do is, by understanding a detailed level of mechanism of exactly what each variant does, it helps us to better define each case of Greer disorder. It helps to better group them together, and so that we can say, okay, you've got loss of function, but it's a different kind of loss of function from that patient, but it's a similar kind of loss of function to that patient. And we can start to have a better understanding almost of the natural history of this, and rather than just having these big lumps, oh, you're all gain of function together, you're all loss of function together, there's subtleties in that, and appreciating those subtleties can potentially be very important in terms of taking the patient down the right road when it ultimately comes to deciding therapies. And of course, as Amy said as well, we're thinking about future clinical trials. And if in such we get to the position where one of these molecules that we're looking at, or a related one, is suddenly in a position they want to have a look in Greer patients, we want to easily be able to tell them, this is the best group of patients. If you want success in your trial, and of course the businesses do, these are the patients who should be in your trial. And that's really, really a really, really important thing. This is all, of course, very, very serious, and Greer disorder is very serious, but I'm a geeky scientist, and I've said this before and I'll say it again. Your kids have some really, really cool ion channels, and I look at all of this. When I look at this analysis, it's exciting to me. So in general, the records in red, these are the different kinds of ways in which receptors can be underactive. Whereas the records in green, in fact, that one's in blue. Ignore that. That's me. That's my laptop on night vision at 1:30 in the morning, making me think that blue was green. Okay, so apologies. Yeah, so they're all green. They should be green. But that one in the top right-hand corner, that green one, it won't mean anything probably to most people in the audience, but 130 picosiemens. It's like this monstrous channel. It's... Yeah. I mean, so Kate Winslet in Titanic said... Dr. Freud had interesting theories on the male preoccupation with size. But I mean, and you read into it what you will. But I love that record. These are the biggest goddamn AMPA receptors that anyone's ever recorded. And I don't care what you think about that, but it's amazing to me. And so basically, I'm enjoying doing these experiments. Your kids have really, really cool ion channels, but because they're so varied, I believe that is going to help us better define these different subdivisions of Greer disorder. And also, I mean, I can't speak for Dr. Train-Ellis, but one of the, I believe the project that's just been funded for him is another way that he's starting to look at these variants to find out, again, another different way in which the variants in GRIN receptors may lead to different ways in which the receptor can be defective, and therefore different ways in which we may be able to funnel a better treatment for a particular subset of patients. So,
Thank you. Okay, and so the same question for Dennis. How will the work lead to treatments and cures?
Yeah, so there's two parts to it. On the one hand, trying to learn the lessons from the basic scientists and try to understand if there's some general rules. So, for example, if you have tested 20 variants and you learn that always the ones in the top part of the channels lead to loss of function, and if the ones which are in the bottom part always lead to gain of function, maybe this is a general pattern. Maybe that's true for all variants which are in the bottom part, which are gain of function, and all the ones which are on top are loss of function. And then computationally test this, and then go and also validate this. So just try to generalize, because, you know, at the end, there can be tens of thousands of possible mutations in any of these big receptors, and we cannot test them all. At least it's very expensive at the moment and slow and so on. But we don't need to. So we could already show this in this left study from Ludovica, that for the GRIN receptors, at least in this one-off static view, which we have at the moment, it looks that there is general rules where the mutation is located and what the corresponding function is. And with more knowledge and more testing from other approaches, we can see if that holds true, if there's different kind of receptor compositions. If there's GRIN2A, GRIN2B, and so on. So
sorry, when you say where it's located, that's where in the gene? Like what part?
Yeah, where in the gene, and that's basically where it's in the gene. It's then also at the end where in the receptor, this molecule which is acting in our body. The gene is always the construction plan, but the active thing which does something in our bodies is the receptor, is a protein. And these relationships from position to function, they then translate then also typically to the version of the disease. And our specific goal for the funded project is really to take the lessons which we have learned for the NMDA receptor and apply it to the AMPA receptor that we don't, so the GRI gene receptor, so that we don't have to test so many variants, that we can use the variants, for example, that Steve or Ian is testing more for validation, and then we don't need to test now 200 variants or so for so-called training, for mapping the thing out. And so that's the project. So if everything works okay, we would be very, so for example, the NMDA receptor prediction model took us 3-4 years, but if for the AMPA receptor model, we might have that already in a couple of months from now. This all comes with caveats, right? So, but big picture, we hope that we don't need to wait for hundreds of variants for the GRI genes to be tested over the next years, so that we can actually learn big picture lessons, more gain of function or more loss of function type way faster, hopefully in this year. And that would have then implications for prognosis and clinical trial enrollment and these kind of things. So this is all an educative guess, right? So this is super important. Even a functional tested variant is also just an interpretation, so nothing is 100%. So that's the one part. And the other part is putting us all again in a portal so that we have an AMPA or GRI gene portal, or GRIA gene portal, also over the next half year, so that people who have who have opinions or are just interested in learning more about the receptor, the genes, and so on, that they can go to a portal which looks like the GRIN portal. Since I think it's unclear if we will expand the GRIN portal and make it a GRI portal, or if this becomes then a GRIA portal separately from the GRIN portal. Open to opinions, I don't care. And so that people who are interested, like families, genetic counselors, and neurologists, industry leaders, that they don't have to talk to millions of people and waste precious time, that they can just go to the website, learn about this, and if they have further questions, then can reach out.
Thank you. So it sounds like really the question that the three gentlemen in the middle are trying to answer is really kind of similar. It's like trying to understand which variants are gain and which variants are loss, and when, in the case of Andy, and then knowing, because that's what we need to know, both for small molecules, especially for small molecules, but even for like whether an ASO would work on a particular patient variant. So it's having that really information about a particular variant to know what kind of current or future medicine could help.
Yeah, maybe even understanding more subtypes, because this gain and loss of functions is at the end already a simplification.
Yes.
And then understanding the variety of maybe strong gain of function, modest gain of function, or maybe different type of function helps then also to understand a little bit drug responses in the future and maybe dosage or other types of related things.
Thank you. Okay, Dr. Dallman, how will your work help us get to treatments?
Okay, so while the slide comes up, I want to say that the project we're doing is really interdisciplinary and involved. So many GRIN2B families have already interacted with Calliope Hollingue, who's also at the meeting, who's doing focus groups to better understand GI symptoms in the community, and that's very important for developing treatments that might be effective. We are also, so part of the project, okay, so then we've got a pediatric gastroenterologist on the team and an adult pediatric an adult gastroenterologist, an app developer, and an app tester. So the app is this Stripes tracker, and it is informed by signs of GI distress rather than verbal explanation of GI distress, so that you can track symptoms over time and perhaps in that way work with your doctors to manage those symptoms. In addition, we are developing a zebrafish model, and we're testing things like how does the GI tract in a GRIN2B model detect nutrients that have entered the gut. So your gut has all these sensory cells that can sort out whether you've eaten like the goods, like the sugars and the fats, and tell your brain, hey, stop eating. The goods have arrived and we've got to absorb them. And that's a really complicated orchestration that can break down. And so understanding what's going on in that process is really important. And the exciting thing is that that field is an emerging field generally. So I think many people have heard of Ozempic. That's based on those sensory cells in the gut talking to the brain. And so there's a lot of, as well as microbial, like you were talking about ketogenic and microbial compounds. So there are many sort of emerging possibilities for treatment if you understand what's going on in terms of the sensing. The other thing is gut motility or peristaltic contractions are really important for moving contents through the gut. I've heard from many families that there is a lot of colonic impaction that can happen. And so understanding how motility is affected is also a really important component that the zebrafish is very useful for because it's just transparent. You can look into it. So based on those experiments, we can potentially test some pro-motility therapies to see if they're effective.
And do you have another slide as well? Let's
there's a
stick
last
with that
slide.
now too, yeah.
So, I mean, and it's really simple. So basically, one of the questions that we have for the community are what are the daily GI challenges and GRI conditions? And I've really, I think we've benefited enormously from coming to this meeting and talking to the community. We understand that a lot better, but I think continuing to work with the community will, through using the app and the muffins, will really help to give us some much better sense of what's going on. And then in parallel, having the zebrafish model, we're hoping that there's feedback between the two to get a better handle on symptoms and how to manage them.
So I gave a little mention of it before, but what will happen at 12:30 if families come to that?
So at 12:30 in Ponce 4, the small room, I'll have QR codes for the app, so anybody can so the way the app is the goal of the study is first to refine the app in collaboration with families. So in the fall, we're going to be recruiting families to help us refine the app to go along with the symptoms that are experienced in the community of GRIN2B. But even in advance of that, we've written the app for the Fallon McDermott community. And so you can download the app. It works by, like, if you track symptoms, that will be stored locally on your phone, like it's yours. You can export the PDF and then interact with your, use it in any way you feel inclined to do so. And that's not limited to GRIN2B. Like, anybody who wants to can download the app if you want to. And so it's a caregiver-assisted tracking, and multiple caregivers multiple caregivers could download it on their phone to track symptoms. So for those that are participating in the study, they would be invited to link to a database that would allow them to participate in the study, that you can engage or not engage. So at noon 30, we're going to go over the app, and then I have samples of the muffins. I have like 14 muffins, which isn't a ton. We're totally willing to cut them up, depending. But yeah, so there will be samples of the muffins. I'll show you some of. I have slides of some of the pilot data so that you can see what I was talking about with the how symptoms change over time. Sorry.
Oh, sorry, I was wondering what the pilot data looked like. Yeah. Blue. Anyway, so that's what's going to happen in Ponce 4.
Okay, thank you. Over to Amy. So the question for you is, what have you learned so far with your research?
Okay, so it's still ongoing. I think that what we've done in the mice, so I have to say that the lead on this is Maggie Kalev at University of Auckland in New Zealand, and she's been doing the work with the human cells and creating kind of the human variants and studying megakaryocytes, which make platelets and red blood cells. But then on our side, we've been working with the mice, and Sylvia Okafor, who, if you donated blood yesterday, you might have met her, as well as Lyra and Emily. And I just wanted to say thank you to the three of you, plus Mara, for doing the blood draws, because they were there all day yesterday. And thank you to everyone for doing that. Okay, so yes, I really am so grateful. On our side, Sylvia has been studying two variants. One of them is a loss of function variant, and one of them is it's been really hard to say whether it's a gain or loss of function. And when we look in the brain, it looks more like a loss of function. But then Sylvia, looking at the red blood cells, it looks like a gain of function there. So it's really complicated. Okay, so this is on the left-hand side. What you're looking at is Sylvia collected red blood cells from the mice, put them in a dish under a microscope, and then that green line is where she applied NMDA to the red blood cells. And when she does this, not all red blood cells have the NMDA receptor on them. So there's a lot of red blood cells that didn't change in response to NMDA. But there are a fraction of cells that do have active functional NMDA receptors on them. So what you see in gray is what the wild type response is. And where it goes up, that means that calcium has come into the cell. So that means that a channel has opened up to allow the calcium in. It causes the cell to fluoresce, and the more fluorescence you detect, the more calcium came in. So that F/F0 means the fluorescence that is detected divided by the fluorescence that you started at time 0. And so, the bigger that number is, the more fluorescence, the more calcium. So you can see that that purple line, that's the patient variant that really looks like a gain of function in the red blood cells. That's a big change. And then on the bottom, you can see what happens in the loss of function. So again, the gray is the wild type, and here you see that the NMDA doesn't really cause a change in the intracellular calcium. So that would be kind of, so what this is, the reason why that's interesting is that, well, getting red blood cells is really easy. Wouldn't it be so nice if we could just take red blood cells, put them under a microscope from a person and say, well, let's add NMDA and see if, we get more calcium or not. And that would be a very simple test. I don't know how useful this will end up being as a biomarker, but I think it does show that, sure enough, there are functional NMDA receptors on red blood cells. And, I think we will probably also find other glutamate receptors on these blood cells. And Maggie's made that her whole living, so she can tell you a lot more about these receptors on these blood cells. So I think this was, for me, convinced. There's a lot of evidence that we have. Sylvia did a lot of other studies that there are these receptors at really low levels on blood cells, which means that it's not crazy to think that we will be able to use these cells as biomarkers. So that's, I think, the most that I can tell you right at this moment, but we're still doing a lot with the samples that we've gotten from Combined Brain, and we'll be doing that with the new samples that we just collected today. We've got quite a few that we can do proteomics on now.
That's great, yeah. And so, just quickly, because we want to get to questions from the audience too, but the focus is on GRIN1. How applicable do you think this could be to other GRI genes? So
I really apologize that I haven't learned yet about the AMPA and kainate receptors, but I know that all of the subunits are detected in the white blood cells, the lymphocytes, but in the red blood cells, 2B is the one that is not detected in the red blood cells. So I think that white blood cells might be, and actually, if you look at the percentage of cells that have NMDA receptors, it's more like 20 to 40% in the B and T lymphocytes, whereas in the red blood cells, it's more like 1 to 5%. So I think we're much more likely to get a better signal if we look at the white blood cells than looking at the red blood cells. So I think it would apply to others, yes.
So just to put it in context for everyone, this is really promising news, because most neurodevelopmental disorders do not have a biomarker in blood. And so if this pans out, it definitely makes our disorders more interesting for industry as something that they would want to invest in. So
Yeah,
thanks for all of that great work. So Dennis, your research is really aggregating different data types. So why is it so important to aggregate and to jointly analyze different data?
Yeah. So where I'm coming from is the following situation. So there's two scenarios where today in clinical care, ultra high risk interventions or therapies are used without complete knowledge if it will work or if you might even hurt the person. So there's on the one hand very cutting edge therapies in oncology, which which really are strongly modifying the biology of the person and with the goal that it targets the tumor, but it can have really severe adverse effects if you give the wrong therapy to the wrong type of tumor. And the other one where I'm a little bit more familiar with is the epilepsy surgery realm. There are types of epilepsy where you have abnormal, really looking neurons, which you can detect in an MRI, and we call this lesions. So you have it in one part of the brain. You see there's like a white thing lighting up in an MRI. And people with this type of epilepsy, they benefit 6 to 7 out of 10 times from cutting it out. So, and what happens is since like 10, 15 years, the ratio of success is only 60 to 70%. Depends a little bit on type of lesion, whatever, but it's 70%. But the procedure doing it, you have to come to a hospital before you get actually electrodes in the brain. You then open the brain up. You go in. You have super high, you have risk for infection, and then you cut it out, and you don't not even know if it helps. So how is this approached? This high risk, potentially not useful, and type of therapy, where the result is really not clear, and every patient is different because these abnormalities are in every person a little bit different, is through the so-called Epilepsy Surgery Management Conference. And there you have experts who are specialized on reading EEGs, and there's even other technologies called MEG, it's a different version of EEG. Then there's the imaging, and then there is the epileptologist who is trying to learn from how the epilepsy looks during the seizure and the history of the patient. And long story short, at the end, there's like a panel of experts who make an educative recommendation based on consensus. And I have been at this for like 5 years, listened to these conferences 3 hours a week at Cleveland Clinic, which is the largest epilepsy surgery center probably in the world. And I learned from this is you can be very proactive in applying cutting-edge medicines, even if you don't know if it will work. But you need to have good ethics, and you need to have a very diverse team on board, all sources of data, and you need to wing it a little bit in terms of combining this information. But if you do this, you can even bring therapies fast to people, but you need to inform people about the risk for everyone, and every therapy has risk. So, and learning from this, I try to apply this a little bit in my research, trying to combine different sources of data, bringing knowledge from different experts in, to then also understand the risks and the rewards, and then potentially giving, recommendations for care. And in my opinion, and I think everyone agrees, is we're living now in a world where the precision therapies are coming. We have physicians who have not been trained for these therapies. We have biologists who have a lot of knowledge, but not in clinical grade. And we are living in a scenario where technology is here, but no one is trained for it. And the only thing what we can do is like sit together and bring all the information together, have a dialogue, and let the family decide, very informed, based on the information which is presented to them, how to move forward. There are certain drugs which have less risk, like molecules which wash out, but the more curative therapies come with very high stakes. And for this, you need to have all kinds of data there is, and unfortunately, most of the data is in research space. And we cannot wait until they become clinical grade. This takes forever. So in that case, we need to bring a lot of people together to make informed decisions.
Thank you. So the most important job of moderator is to keep everything on time, and I have not done a good job, but because we're at time now, and we still have one last question for Ian and Andy, and also hope to still take maybe one question from the audience and one from online. Who's the mic runner for the session? Okay. Hello. Okay. So, Ian, while we're doing that, can you tell us how close we are to therapies for AMPA receptors? In 2 minutes.
Okay, I will. Right. Okay. So I took the risk again of being sued. I've put some Disney pictures on this slide. Oh no, they're gone. So, somebody said, cut. We can't afford a lawsuit. Okay. So I guess I already showed a little bit of data in the first slide that we are. It is certainly possible these molecules exist that can modify the behavior of GRIA variants to kind of normalize them. They're not make them perfect, but then potentially undo lots of the variant. But as I said, until these drugs are actually available in the clinic, then it's kind of like it's good to be ready, but we can't really plan for it. So I guess what we're excited about is just this year, for the first time, one of these selective AMPA receptor PAMs that has a big effect on the AMPA receptor has made it into a phase 3 trial. And this is like this is where we're really starting to get close to something that might be approved. Maybe there's some Montanucci who can fact check this. As I went on the internet and I had a whole load of mathematically impossible things that all came together, and it told me a percentage of how when something goes into phase 3, how many drugs come out the other end and actually get approved. I've averaged this really very wide range, and it came out at potentially about a 25%. I mean, I don't know. I can see some Novartis people in the. Am I even right close to this? At once the drug's in phase 3, maybe 25% of them actually make it through successfully. Is that an overestimate, an underestimate, or?
Yeah, I thought it was more like 50. That's in the U.S. anyway, but.
Oh, sorry. It should be more than that. Oh, great. Okay. Well, that's good news. Okay. So typically we're talking 2 to 5 years, and at the moment there's a molecule that's in trial for depression, and so you need to have something being tested against a big disease to make the money that's going to be worth the investment for the company. And the key is that if that comes out and that gets approved, then it would be possible, far more easy, to get that drug into GRIA patients and GRIA loss of function patients who currently don't have. So when I have my perampanel for you, no perampanel for you, I can now say perampanel for you, drug X that's soon to be approved for you, and it kind of it gives an extra thing that potentially some of the GRIA patients can try. I won't go through the rest of it, but I'm happy to talk about it if you want to catch me, I guess for time. But there are other molecules out there as well, some of which are not really directly selective for AMPA receptors, but do seem to have some effect against them. And again, these are some of the molecules we're looking at, and we're hoping to provide support for the idea that some of these molecules might be useful for some GRIA patients in the near future. So that's where I'll finish.
Okay, I'm sorry to do this to you, Andy, but can you do it in 1 minute? Your last question, which is how does your lab work? How do you do your work?
Ian.
Sorry.
Right, so...
Don't wait for the slide.
No slide, okay. Right, so the way that it's, not this slide, it's the next slide. So our lab is looking at that window where, around birth, taking cells out of mice, particularly neurons, and we grow these neurons in a dish, and what we're doing to make sure that we can really answer this question and be able to look at how the relative amounts of GRIN2A and GRIN2B are affecting GRIN2B variants. We really want to properly replace GRIN2B in the mouse with the human variant of GRIN2B, really expressed at the level that it should be. So we've applied for the first time a CRISPR knock-in strategy for doing that, and it allows us to put in the GRIN2B variant and identify those cells that absolutely have that variant, so that we can then record for them and look at how the function of the receptors at the synapses is changing in development. And actually this morning at 7 o'clock, I got an email with a wonderful picture of a graph that shows data from 375 cells that shows that this knock-in strategy is working really nicely at about 45%. I would love to have put that in my slides on Thursday, but I only got it this morning.
Okay, that's great news. That's great to hear. Okay, I think we have one question from Ashley.
Hi. So I'm interested in knowing what is on the horizon, or if there's something on the horizon regarding treatments for these children that fall on the border between loss of function and gain of function. There's so many parents that I come in contact with that don't know how to help their children because they haven't been given any sort of data about where their child actually falls on the spectrum. And I feel like there's actually like quite a group of these children. And so like, is there a plan? Is there something on the horizon to help these kids?
What comes to mind for me is that a gene therapy that doesn't care about what the variant is, right? So either to edit it, to do an antisense oligonucleotide, or even a gene replacement strategy that is not, that can work either way. So I think instead of, I mean, anything that works at the DNA level, it's not about what the substitution is or whether it's a truncation. It's more about correcting what, the problem is. So that's what comes to mind for me.
Yeah, I can just add that the type of, the thing I just showed on the slide using CRISPR, I mean, we're doing it to make the variant in the mouse, but you could do it the other way around. And so what we're learning from using this tool, the way that we're using it now, could be a way that we can do this sort of thing like that Amy was doing. Although this project isn't designed exactly to do that, it will help us understand the tool better and the limitations of it to know to what extent we'll be able to apply it in the other way around. So.
Yeah, and so the good thing is this year is actually the first time where I personally observe in the very top clinical journals that these editing strategies are now more frequently tested by hospitals and which work together with scientists in these more like N of 1 studies. So there's probably, to the best of my knowledge, no specific GRIN program for editing, specifically on large scale out, but there's a lot of cutting edge hospitals which really try to push the boundaries, which work on specific selected patients. I cannot speak to the selection process, but this is something for this community where the function is unclear to work proactively with the organization and map out these hospitals and investigators who are willing to explore this and see that you can become part of it.
Yeah, I mean, Stanford's a great example because they've done a custom CRISPR patient disorder, and they just got a grant from Chen Zuckerberg Initiative to kind of expand the work that they're doing. But there's others as well, so that's great. Thank you to the panelists. This has been a great conversation.